So I've just highlighted some points from each of the speakers. So we've heard today about affinity purification, followed by mass spectrometry to separate, as Rob Ewing called, the wheat from the chaff to characterize pathway players, particularly in control versus phenotype. We've heard a little bit about, from Mike Washburn, about spectral counting to assign confidence scores to interactions, and then how he forwarded that type of approach to looking at deletion network analysis, which I found very interesting myself. We learned a little bit about affinity purification, mass spectrometry to identify gene regulatory complexes, and how we can use MRM experiments and cross-linking experiments to identify functional interaction sites, which leads to confidence building and interaction networks. And that was brought to us by Jeff Ranish. Danny Schlatzer in our center talked a little bit about how we can use peptide markers discovered in human disease and map those to an interactome to come up with maybe candidate subnetworks that can be the basis for follow-on in vitro experiments and in vivo experiments to test the relevance of those networks mechanistically in disease. Anne talked to us a little bit about building out the physical PPI maps to extend knowledge from genetic interaction networks. After that, we all enjoyed a nice lunch, and then Akalish talked to us about phosphoproteomics in disease and was emphatic about reminding all of us that expression, differential expression, does not equal activity, which is something I need to be reminded of when we start using models for looking at differential activity between control and disease, that it's not just the expression, but we have to integrate other data types as well to improve our models for activity. Then Alexei reminded all of us of the quantitative rigor that underlies peptide and protein identification at the level of database searching, and how that can lead to improving confidence scores of interactions. Mehmet talked a little bit about computational methods for identifying significant subnetworks using Interactome databases in disease. And then finally, Gary talked to us about interaction prediction across species. So we've compiled 3 questions that we emailed in advance to each of the speakers, and we're just going to go through them one by one. Okay, so the first question is, and Mehmet, you've got the mic there. So what, in your opinion, are the most important challenges in obtaining a complete and accurate map of the human interactome?
I don't have much to say about the experimental challenges. I think, by time the experimental challenges will be overcome. But at the moment, in terms of using what is available, that is the somehow modeling and representing what we obtain from experiments, I don't think we will be able to go to where we want to go with simple binary network representations. So I think in terms of computational modeling of interactions, I think one important obstacle is the binary representation of interactions. Simply, having proteins and representing their interactions in between with a simple edge. So it can't go far. We have to come up with novel modeling paradigms. People have to understand the differences between forms of interactions. Being involved in the same complex is different compared to signaling to each other, etc. So we have to start annotating the interactions. So I think being able to do that will really make the human interactome more useful in terms of understanding biological processes.
So I can make 2 brief comments more on the sort of experimental side. I think firstly it's extremely important to sort of define the system. So by that we need to figure out which interactome we're actually trying to map, because obviously you're going to have differences under different cells in different states and different tissues and all the rest of it. The second point I'd say is I think we have a lot to learn from other communities like the genomics community, where it became quite clear that single pass coverage of any genome sequence was not sufficient. So at what depth do we have to map our interactions in order to ensure that we have sufficient confidence in them. I think there's a lot of technical things that we need to work out, methodological things, so that we can really begin to say exactly what the problem is and how much work it is to achieve this.
Yeah, so the question is, what are the important challenges in obtaining a complete and accurate map? I feel that I'll go out to an extreme, and I would say that, first of all, I think we may never get it. To admit the possibility that we won't have it. And I think we are setting the stage up to try to get what some people in the community may call a complete or comprehensive coverage. And I think right there is a problem. And I think some of the issues that Gary Bader is finding in terms of not being able to get the signals that he needs in order to predict interactions in what I would say some of the best studied protein-protein interaction systems on the planet, is because they're still not comprehensive. And that is really the kinds of protein interactions that I would argue that people have beaten to death. And we have data from many different platforms. And I think there are lots of challenges. Still stepping back and saying that, you know, we can accept many different intermediate solutions along the way. I would say that the challenges are that we don't have a complete collection of baits and tools. So we need baits, so they can be tagged molecules for certain kind of studies, and tools like antibody type reagents. So somebody could argue, I would argue that yes, we need to look at endogenous level of proteins, but then we need those antibodies, and the other kinds of initiatives have to keep up, and we don't have antibody type tools. I also feel that at some point everyone is trying to sell their technology. There are many different platforms. Some people think of yeast to hybrid as the only platform, AP, mass spec, but then individual experimentation in different labs. I feel as a community, it would be nice if we could do some sort of a divide and conquer approaches. But people are competing actually for funds in many cases, which is another challenge itself. If funds were unlimited, then I think people would feel more secure and do things a little more systematically, or we as a community. There are also technical problems in getting this complete coverage. Some proteins are just very, very difficult. Many of these things, the reason SH2, PDZ domains, they were worked on first is, you know, they were easy to work with. Early on, people figured out they're nicely molecularly defined interactions. SH3 domains bind such and such a protein. You could design peptide libraries. For many of the others, it's not intuitively obvious. If you have a 7-transmembrane protein, what kinds of proteins would bind? So it's difficult experimentally, and we may get much less coverage for those kinds of things. And different platforms may be needed to get an accurate picture, and the expense of the experiments. I think, like I was saying earlier, that one may need to do this in many, many different cell lines under different contexts. And that's why I'm arguing it may never be done. It may be much below our detection limit if we have to look at primary neurons at a given stage of development. And yet we are arguing we need to have a complete. That's complete maybe for, often sometimes I feel, for computational purposes. Right. That's why we are rushing, but I think for a complete biological perspective, I think it's not going to be that easy. But these are challenges enough. I think they are at every level that we can imagine.
Complete may be a little difficult to understand when you think that the interactome is probably changing under very different environmental conditions, under different pathological conditions. Things get post-translationally modified, so just the combinatorial problem alone prevents us from ever knowing the complete interactome. So what I'm hearing is the problem is money, it's technology, but it's also cooperation amongst researchers as well. And maybe that can be alleviated somewhat so we can move in that direction.
Well, I think you covered many of the points that I would like to say. Of course, it's going to be we should definitely try to do this. We have to do this. And I think we should start in maybe a defined cell line or cell system where we can produce enough of the material to start to get at this problem. We need methods to efficiently tag all these genes. So I guess methods that, or let's see, you were mentioning earlier, those knock-in approaches to comprehensively tag the proteome, and then methods for purification of all the proteins, and then identification of potential interacting proteins. I think figuring out what actually the protein-protein interactions are within these purification schemes is going to be really important. So like techniques like cross-linking and maybe integrating yeast-2 hybrid kinds of approaches to figure out what actually the direct protein-protein interactions are going to be really important to get at this problem. Then dynamics and cell type kinds of problems, that's going to take a lot longer, and I think we're going to have to hold our breath for that for now in terms of completeness. Those are the main things.
From the computational perspective, I mean, it seems obvious that not a single method would be able to get a complete and accurate coverage. That's how you define that. So that means that if there are 10 different methods used in parallel, so there are got to be 10 different computational models for each different data types that are tested and validated. And then on top of that, integration of these different subsets of Interactome integrated on a high level to get a complete map. So that's from the computational data analysis perspective, that's going to be really challenging. I mean, this is experience from all this type of studies, that at the end, the data analysis takes longer than data collection. So if you feel that it's challenging enough to collect data, it's probably going to be even challenging to actually analyze it at the end and get a high quality data set.
Pretty much everything's two things. I agree with Akilish, and it's the question is it even worthwhile to go for and to use the word completeness, because it's probably not attainable. And the only other thing that I would add, being at this part of the line, is I think that NIH has a complete and utter lack of vision, and they're clueless. I don't know how to put it any more bluntly. I think they're just doing an absolutely terrible job when it comes to this type of analyses. They're doing all of these huge genomic studies. They have all these orphan genes that no one knows what they do. And the technology exists to pull all this type of things off. And I just think that in this country, NIH is so far behind, say, what Genome Canada and the European Union are doing, that we'll always be third.
So, yeah, I was going to agree with everything everyone said before, but no, I'm kidding. The only thing I could add, I think, is that we have to obviously we need a huge amount more information, but once we get that information, if we're able to learn some general rules or models, then we'll be able to think more about completeness, even though we haven't experimentally mapped everything, but you may be able to have a general model where you could use that to understand the effect of something in a system that you haven't yet measured, just like they do, people do in physics. And that gets us more towards completeness. It's not complete in terms of the map, but it's more complete in terms of understanding. So that's one. Obviously, we need a huge amount more data before we get there, but that's one way that we could think about completeness.
All right, so unbelievably, I still have things to say. Two things. The first thing is, I'm surprised that didn't come up today. I think, especially in the case for APMS, it's clear that it has to be a community effort, and many labs are going to be involved. And so we have to work as a team, and we have to benchmark each of the methods and each of the pipeline using good statistical tools, good control data, positive and negative data sets, so that we know and know how to merge all of these data sets so that we can get to a map that is of some level of quality. The good news is, if I look at the recent data that has been published on a similar system than what we're working on, and same thing for Mike, I'm sure, is the data quality in many labs is going up a lot. So I think we're very close to being able to pull this off. Second thing is, in terms of what else do you need? What else do you need beyond 2 hybrid, beyond maybe protein fragment complementation, beyond APMS? Is you might need things that you don't think about normally, and I came to the realization of that recently, thanks to Charlie Boone, that has been doing genetic interaction in yeast, right? So it's doing really high density genetic interaction, and the density was high enough that looking at the profiles of the genetic interaction, it can actually predict protein interaction that were not detected by any of the other techniques before. So, and we see something very similar coming out of siRNA screens now, where we have a bunch of proteins that have the same phenotype, and then we go back with more sensitive and directed approach, and in fact, we can show protein interactions. So, you know, beyond the standard ways to detect protein interaction, I think we need to stay in tune with these other communities doing genetic information, both by siRNA screen and other techniques in SGA, because really that fits really, really nicely and complements what we're doing.
So I was going to say something provocative, but first I would like to add maybe to the list of all kinds of limitations, basic limitations, things that go beyond even combinatorial problems with protein-protein interactions, and somehow they never got mentioned. So certain proteins will interact with other proteins only in the context of DNA and chromatin structure that would involve some very non-local effects due to chromatin modifiers. Some other proteins will interact with yet other proteins in the context of membranes. Again, with some very non-local effects, sometimes because of membrane curvature and so on. Some proteins will interact with other proteins because of crowded environment here and there in the cell. So there are all kinds of very fundamental limitations that go into the direction of combining all these beautiful efforts with other approaches. But the provocative thought that I was actually going to mention is the following. With all this beautiful work, you can tell that whenever you look at some system more carefully, you find many, many more interactions. That's sort of general conclusion from some of today's talks. And one could argue that maybe right now we know at least 10 interactions per protein on average. There are some that have 150, 200 known cofactors and interactants, androgen receptor and other things. And one could say that, well, the most predictive factor in this is just the focus of and how many people study these interactions. So if we were to predict the number of interacting partners as we know them today, perhaps paradoxically the most predictive feature would be how many people work on a given protein. And as we progress with all these studies in the future, we'll learn more and more about all these interactions, all those interactions that have not been detected as yet in different contexts and so on and so forth. And as we go towards the complete map of Interactome, maybe we should just stop now and say, with time, if we consider all possible context, isoforms and everything is going to be related, any protein is going to be related to any other protein with, say, 1 degree of separation. So that's my provocative thought. Maybe we could just say, well, everything is going to be related to everything.
So these two hybrid people have found like that to basically not be true, is that there's orphan space that always remains orphan space, where because no one's interested, no one ever goes after it. And so they push really, they've had a recent publication on, you know, they think they've done 10% or something along these lines by estimating it. So I believe that that won't happen. They've been addressing those types of issues. The point being, though, is if you just rely on the individual interests of people here and people around the world, there will be certain parts that, like, it's still to this day in the yeast affinity purification. I mean, Jack Greenblatt's working on this, but it hasn't really been attempted to do all the membrane protein interactions. You'd think that would be the place to do it. So there's hard problems out there, and there's areas of biology and proteins that you know aren't, or genes that you know are important, but no one's studying them for all sorts of reasons, most of them being funding type of reasons. So, but those spaces to get complete maps you have to do, and someone's got to be willing to do it. And it won't happen if it's just people studying transcription, kinases, and phosphatases. There's a huge part of metabolism, and there's aspects of all sorts of cells and cell types that will never be addressed unless you say, all right, we're just going to go for these things that we know nothing about. So I think it has to be done in, you have to be willing to address these areas that no one is willing to, no one's touching at this point in time.
I have a similar question. It's kind of an extension of what he said. In any of the models, in any of the peptide predictions that you have, do any of them, first of all, predict and have been confirmed with the, like, self-self interactions. In other words, you pulled out all those peptides. Do they match for like when you have a homodimer or a homotetramer? Do you find the same peptides and say, okay, this protein now will dimerize, and then you go and confirm, say, oh yeah, it does dimerize over there, or it does tetramer. And the second point is, I'm interested in gene-nutrient interactions and nutrition, and a lot of disease is not simply the genome. And I'm also interested in like metals. For instance, you could have hemoglobin, you could check it out, and it'll interact with this many proteins. But once the heme goes in, depending on its oxidation state, it's going to interact with more proteins and different proteins, or NOS, or the zinc fingers. Do any of the models predict those kind of interactions?
Somebody have a quick response? Ann? Gary?
Well, the dimerization can occur, but it doesn't happen that much in the ones that we're looking at least. So PDZ domains can bind each other sometimes. But I guess the second question, anybody here could answer.
The current model do not modelize that, though.
The current models are not, yeah, so that's a goal to add that information in, but that's actually an aspect of, maybe that's another aspect of the complete map, getting to the dynamics, looking at protein expression levels and all these other measuring molecular profiles at all different other things like the epigenetic state of the cell, or all these different things that people can measure on a global scale. Obviously those relate to the dynamics, which ideally we need that information as well, and hopefully that information could be put together with the protein interaction information.
The second question posed was, how does the relatively low coverage of the current Interactome maps impact their utility and our ability to mine these data? So some of the recent estimates of the extent of the human Interactome are maybe as low as 1% or 2%, but they're awfully small. So we're asking you to comment on what that low coverage estimate, how that impacts our utility and ability to mine these data. So Ann, why don't you start?
I think Gary and other people would be better to comment on the computational aspect and predictive aspects of this, which is not what I do. At the experimental level, the low coverage is pretty much telling me I need to do the experiment again. If I am interested in a gene that's implicated in disease, and I've shown a couple of slides of that, everything we've found essentially has been new. So we've been doing a lot of experiments now looking at disease-specific alleles, or, well, I wouldn't say a lot, but we started doing disease-specific alleles and other mutants that may be affected in their disease outcome or cellular outcome. And we find that we have to repeat the experiment by ourselves to be able to map systematically the interaction profile. So I think many people doing the same kind of work that I do are starting to do this kind of experiment, and clearly the Vidal lab just got, I think, 2 papers in press in Nature Methods and some other journals that deals with that in kind of a much higher throughput. So I think we're still at the beginning of this kind of usefulness, and for us it's been meaning go back to the bench.
Yeah, I agree. I guess with the Human Genome Project, it wasn't finished until it was 7 or 10 times complete, done over, right? So you need to do that. I guess if Akhilesh is right and it's never going to be complete, then even though it's demoralizing thought, we're actually then at 0 right now, no matter what we do. But maybe that's just too demoralizing.
I have a simple answer to that question, just 2 words. I think the answer is huge and bad, where it has a huge impact and we really can't make many predictions because we just don't have, we have so little knowledge.
Yeah, I guess low coverage also means low confidence. I mean, if you collected your data set and it's very low coverage, you compare with some other data sets on the same system, and there is a 0 overlap, so you don't even know if you trust even those few interactions that you have in your case, right? Because they don't overlap with anything else. So maybe they're true, you profile different subsets of the same space.
Yeah, I think the lack of information that we have is really limiting, and we really have to make the measurements to expand our knowledge about this problem. Measurements.
Yeah, I think because there is so little data, I think people don't have faith in the existing resources. They don't look them up enough, although there's still substantial amounts of data. And I think we need to generate a lot more data. And that's where I agree with Mike, what he was talking about. NIH is short-sighted because, I mean, we are not talking about saturation. For the data to become usable beyond, and that's why I made that comment, beyond the computational field, Computational people, I think, have enough data just to start off. For the biologists, for us to impact their life, it has to be in their context. And right now, many of the proteins, like you're talking about, are completely underrepresented. It's certain communities that work in certain things, and for some reason, the reason that we are having this discussion is somehow these core, centralized, high-throughput proteomics type labs are supposed to do it. I don't think that's my divide and conquer. I think every single person, if you work on endothelial cell biology, you're the world's expert on membranes in endothelial cells from arteries. That's what you should be doing, and we should have funds for those kinds of people, and then these kinds of data sets. Right now, what we are generally sampling is more abundant proteins, more common interactions in certain cell types that the initial people have set already. HeLa cells, 293 cells. So if somebody is going to make an exciting discovery in B or T cells, they're finding some missing because, boy, 293 cells and HeLa cells are not those cells. They will not have faith, they will not come to those. And of course, then we talk about dynamics and context and development and all of that. And we're still arguing human. I think many people who are actually, conversely, we can say people in yeast may not learn so much from human, just the way the other way may not be always true, and then so on and so on about Drosophila and flies and all of that.
Mark, I want to jump in because I think that's the answer. I think the technologies have to be made easier. They have to be disseminated into a wide range of laboratories so people who are experts in a specific domain can do APMS in their laboratory easily, often. That's how, with their 20,000 PIs, okay, who can dig into all of the domains that they are interested in, then it's going to be an entirely different landscape for this kind of technology. Think of how fast genome sequencing has moved. Think of what we dream about. $1,000 genomes are not that far off. You know, I remember but I remember very distinctly when the experts told us it was completely impossible to sequence the human genome at all. I agree with you, there's got to be investment, but there's a very talented, sophisticated group of people up on that stage, and I think they have to start making their case, our case, in a little more coherent kinds of way. I mean, we had the discussion at dinner last night about Wolstein and Markowitz, who are, you know, the Hopkins case colon cancer gurus, and it's genome, 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 genome. And they're starting to realize, man, it ain't going as fast as I would like. And I think we have to make the case to them, they need to understand their phosphoproteins. They need to understand their pathways, and we'll start to inch forward. I don't know that we're going to have a revolution tomorrow, right? But again, if we don't get there, if we don't disseminate those technologies, if only big core facilities are going to do all of the experiments, you know, the big core facilities need to move ahead. You guys need to be doing none of the things you're doing now in 5 years because you've moved so far ahead. And everything you do routinely now needs to be out in the laboratories and the community in 5 years.
We need a lot of education of the genomics community, and it's not trivial what you just said, that Francis Collins is the head. I'm in the Institute of Genetic Medicine. My goal was 7 years ago, I thought I would be the person doing, because they all have mutations in their favorite genes, how they interact differently with different proteins. Not one person is really interested. We can talk about papers people will publish in Nature Genetics and move on with their lives. That gene, that mutation causes, and so much money is being spent on genome-wide association studies. We are going full blast in one arena where it remains to be proven what exactly it's going to do. And the other thing is, it's just ignorance. It's so sad that we are not talking about biologists where people need understanding of genome, transcriptome, proteome, cell biology, and everything else to see how a cell functions. But people say, I'm a genomics expert, I just do this, and this is what I need money for, and they get tens of millions of dollars so easily by replacing fill in the blanks. I'm looking at this gene in this population, and here we are going to just discuss, but nothing will happen. I don't think so, because many of these people are beyond hope, honestly. Seriously, they are beyond. I have struggled. My prognosis is very grim. Canada, I think, hats off to Canada. They're into proteome initiatives, and in the US there are some which say biomarker and this and that, you know, where it is coming together a little bit, but again, hype is ahead of where it is at, 2 years later they're going to say, oh, it didn't deliver, so let's go back to genome again. We tried proteome.
The only thing I would add, I just sort of thought of a conversation that we had last night about, if you like, the proteomics advocacy, if you like, society, i.e. HUPO, and how ineffectual it was. And I don't know how many people are familiar with HUPO, but a number of people here, I think, were at the HUPO meeting a couple of weeks back in Toronto. And from all the people I've spoken to, people did get this sense that HUPO is not going to be the organization that actually drives this movement, going back to the things that Mark said and Akhilesh has said, that really drives this forward. So if that's not the organization that's got to do it, it's got to come from the scientists.
So one thing I would like to add is, since I have been in this field, I have observed many times that rich is getting richer and error is getting propagated. So I think this has to do with the incompleteness of data. And also, I just have probably 10 years of academic life so far, but when I started, I trusted the papers I read more. I trusted the data more. I mean, in 10 years, I can feel that things are getting worse because everybody is just trying to get easy access to data, try their ideas on the data quickly, publish it, go forward. So I think somehow policies have to be developed to make sure that to reward high quality and reliable science.
Has anybody run across protein glycosylation or discussed the effects of protein glycosylation? And then the other aspects are the discussion that you're going through right now sounds just like what the Glycobiology Society discussions are about how we're ignored and no one understands us and we're not getting enough funded and we have to get more people on the study sections at NIH.
I always say, Jerry Hart is my chair, and I say that the people who work on carbohydrate sugars, they're the sweetest. They are the sweetest community. That is the only one community that I have seen where they feel so ignored. They want to be all-inclusive. They always want their neighbor to work on carbohydrates. In many other communities, people say, oh my God, everyone is trashing everyone else's method. Everyone is just downsizing what the community is doing, don't want other people to come in, and I think that's just the wrong model. But the carbohydrate community, it's really amazing that they would like others to join because they realize that problem, again, is almost unsolvable. So the more human beings we have, the more people who burn the midnight oil, the faster we are to finding a solution, analytical, biological, or otherwise.
Specifically with respect to mass spectrometry, I know that there are certain people who are, because of this electron transfer dissociation, ETD is apparently turning out to be very useful for glycosylation analysis. So I think it's having a resurgence. Say, for example, Don Hunt has apparently moved away from phosphorylation by ETD towards glycosylation. There's a very good person at the University of Kansas who does a lot of glycosylation mapping. So I think that there's technologies now in mass spectrometry that are making it more amenable to being used by, it won't happen in my laboratory, but by people who use these kind of same technologies.
Number 3, how can the interaction networks be used to understand the molecular basis of human disease?
Well, I think I kind of covered my answer in the previous one, but I think this is a tremendous potential right now because we know much more about the mutations that are implicated in the disease. In many cases, we have decent cell models to bring back the gene in, and then we can build the interaction network for that. I mean, just in my small lab, we stumble upon several things by chance, and our collaborators certainly are doing so. So I think this is crucial. I think this is going to be really the next phase for interaction proteomics. Once we figure out all the big machines, how they work and stuff like that, then I think it's great.
Yeah, so I think that it's vital to have this information to understand disease because you're running out of these single gene, single disease, these easy to find associations, and everything. The geneticists now they have bigger bigger sets of markers to work with, so they're finding more. But in the end, there's going to be interactions between genes, and you only have and environment and other things like that. But so interactions will help you understand that, and pathways will help you understand that. I think Mehmet had a really good talk today about, approaches like that will help you do better at these association studies. So the interaction data is going to basically be required to get at that level in using methods like what Mehmet talked about.
Just personally speaking, we're really excited about the dynamics and the kind of stimulating or manipulating networks and reanalyzing them, like these deletion networks we're finding to be incredibly informative. You can imagine looking at complexes during the cell cycle using the technologies. You could look at drug treatment type of networks and cellular comparisons of, normal versus cancerous cells. And if we have the right reagents, say like a massive lentiviral library for all human proteins that probably will exist someday, you can start to do very interesting dynamic analyses, and that almost always gives you functional insights.
So my primary appointment is actually in pathology, and so the fact that a person like me, a computational proteomics person in the clinical department, already speaks to the interest in using proteomics in the context of disease studies, but not for global profiling. But I just know 5-6 groups in our department that before knew nothing about proteomics. They all found that analysis of protein complexes is extremely useful for whatever they're doing, and it's usually a different group is doing completely different things, but kind of a common ground is that there are a few interactions that are absolutely critical for the things that they're studying, and the IPMS type of protein interaction analysis is a really important component to that effort. So I think there are many different applications. It's hard to say, which one is going to be the most useful, but there are a lot of useful things that they can do using this approach.
Proteins are crucial for carrying out normal cell functions, and when protein functions perturbed, it can result in really devastating diseases. So it's just obvious that we have to really have a firm grasp on the protein interactions and how those protein interactions regulate the activity of proteins, both in normal and disease states. So it's just, it has to be done. We have to do it.
Yeah, many of these genome-wide studies where, a lot of money is being spent, genome-wide association studies, mutation finding studies, we are looking at cancer genomes, and when the cancer mutations cannot explain the phenotype, then we say it must be pathways and networks. So I think the community is slowly coming to it. Exactly how they will get to it, I think, is because there is less data, I think there are less success stories. And I feel that at the end of the day, we are like sheep. We have many, many examples. We are going to say that is the obvious way. And because we are so poor on the data, that is, we even getting started off the block and going towards being comprehensive, we have fewer success stories. But what the genomic community is really struggling with is once they get to a 10 megabase region, or 1 megabase region, or 100 kilobase region, they have a SNP that doesn't often tell you that this is the gene. They look in the neighborhood, they have no idea what to target next, what to study. And I think this is where not just interactions, but understanding the functions of proteins in biological processes, and I'm not talking about gene ontology terms now, in actually what the proteins do in the cells, I think that will go a long way. And what Mike talked about, these all types of perturbation experiments will basically get us to understand what is a given protein does in a cell, so that that data then can be mapped onto the genome or onto the mutation to figure out how it is behaving in that abnormal situation.
The only thing I would add is sort of reiterating the point that Akhilesh made earlier in the session, in talking about having using appropriate models, disease-relevant models, and I guess I feel that sort of applies to people like me who are using standard cellular, sort of cell-based techniques that maybe don't have immediate disease relevance. I think our first and our third questions, the goals are to some extent a little bit orthogonal. They're synergistic as well. But if you're going to have a complete map of a model system, that's not necessarily completely compatible with really going after disease things. And I think it just brings up a discussion, actually going back to the HUPO meeting, between sort of proponents when they were talking about this complete interactome map of a gene-centric or a disease approach. There's got to be some balance there.
I believe that in disease modeling and understanding disease, the big leap will be if we can actually enable in silico perturbation experiments, if we can save mice. Then I think that will be a big progress. But I think for that to be achieved, people have to define what they mean by network mechanisms. You always see people talking about network mechanisms of disease and etc. But what I see is there is the qualitative modeling of the network, which is simply mostly about who is related to whom. And then there is low-level quantitative modeling, which is probably overly detailed for our purposes. So the network mechanisms are defined somewhere in between, but I don't think people still have answer to what network mechanisms mean, really.